Bismillahirrahmanirrahim. The topic of today is the cells and the tissue, and this is the chapter number 4, in which we will start our course from the microscopy. The first topic of this chapter will be the microscopy. Microscopy is a field in which we study the living organism under the microscope, about those organisms which are microorganisms. Microorganisms are those organisms which cannot be seen with the naked eye of the human, and for the study of which we need microscope, and the field is known as microscopy. So here in this chapter, first of all, we will discuss the microscopy, the different microscope, and the way of the study any organism, about their different features, and then we'll go through the cells and then the tissues in this chapter. First of all, when we use the microscopy or when we use the microscope, we must know about two things. Number one, magnification, and the second one is the resolution. Magnification is the increase in size of any object. The object which we use in the microscope, we increase the size of that object, and we can only see it when we increase the size, and for this the term magnification is used. While resolution is the measure of clarity, that how much the image of that object is clear to us. Sometimes when we magnify more an object, it becomes blurred, but when we clearly see the object under the microscope, it is a special resolution. For example, the human eye resolution is 0.1 millimeter. It means that if the two objects are apart from each other, the distance between them is 0.1, human eye can can clearly see it. And if the distance change from 0 to 0.1 to any other figure like 0.2, 3, 4, 5, it means that the human eye cannot be resolve it or can clearly see it, because the resolution is 0.1. So below 0.1 and up to 0.1, human eye can see. So that is the resolution or the clarity the human eye. Now we will study about the different microscopes used in the microscopy. In your book, the two types of the microscopes are given. Number one is the light microscope. Light microscope are those microscopes in which we use the light as a source of differentiation the object. To see the objects, we use the light. The light must be maybe the sunlight, or sometime it may be the tube light or torch light. Any source of light can use here. These microscopes have two lenses. One lens produces image of the specimen of the object for which we use the microscope, and the other lens magnify it and project it into the human eye. Sometimes the special photos are taken from the microscope, and they are known as micrographs. The micrographs are the photographs which are taken from the microscope. The light microscope magnification is 1500x. It means that it can enlarge the image, it can show the enlarged image of the object 1500 times more than its real size, and its resolution is 0.2 millimeter, that it can clearly show us an image of 1500 times bigger than normal, and clearly it can show us an image that is 0.2 millimeter apart. The other microscope is the electron microscope. As the name mentioned, here the electrons are used. Electrons are used as a source of producing image of the very nano or very small objects. The characteristic of these electron microscope is here. First of all, electrons are used as a source of producing image. The second one is the uses of the electromagnetic lenses. Electromagnetic lenses are present in a vacuum chamber, and when electrons are passed from the object, from the species or specimen, that electromagnetic lenses then produce image of that object on a large screen. The magnification is much more, 250,000 times more than light microscope, and the resolution power is 0.2 nanometer. The so small organism can be seen or detected by using electron microscope. There are two types of the electron microscope as mentioned here, TEM and SEM. Transmission electron microscope and scanning electron microscope. Transmission electron microscope are used for the study of the internal structure of the cell. Internal structures, parts of the cell, like different organelles, and here the electrons are transmitted through the specimen. That is why this is known as transmission electron microscope. While the scanning electron microscope, the electrons are reflected from the metal surfaces, and they are used for the structure of the cell surfaces, means any material that is present outside the cells can be studied through SEM or scanning electron microscope.